Thank you very much for having me here, and which is a wonderful excuse to be back home in certain ways. My lab is very interested in the mechanism at the basis of epilepsy and cognitive diseases, and we think the two things get together, and I will try to do that in this presentation as well. We are physiologists, and the physiologists tend to be totally incomprehensible. I mean, because of the mental deficit that they have, quite obviously. So I really will try to make clear at least what is at the basis of our work, and please ask questions even during the talk, I mean, or afterwards, or throw me something. I would be happy to take anything. Our work is based on a conditional mouse for PCDH19, which has been developed in Milan by Silvia Bassani and Maria Passafaro. We have been collaborating for many years during the design of the mouse, and now finally that we are doing the physiology. And most of the experiments I will show you have been performed by Silvia Landi, Didi Lammers, and Vinocene Pillai. So what I'm going to talk about? I will spend a few minutes to talk about the model of PCDH19 that we're going to use. And I will talk a little bit about the basis, certain ways of certain phenotypes relative to hyperexcitability and epilepsy, which is this dialogue that there is in the central nervous system between excitatory neurons, which are the neurons that make us moving our arms and to have an output to the world, and inhibitory interneurons, which is a much smaller classes of cells. They represent about 15–20% of the neurons in the brain, but they are essential for the control of electrical activity in the brain, and without them we would continuously having epileptic seizures. The two systems interact in a very beautiful and delicate way. We don't know yet how this happens, both the neurodevelopmental mechanism that brings about this dialogue between these two systems, and how this dialogue is regulated during postnatal life, during memory and learning, and so on. Then I will show you two different, but I think strongly correlated phenotypes we find in our experimental model. The first one has to do with sleep disturbances. We find a alteration of a very specific pattern of electrical activity that occurs during sleep, and I would be extremely interested to have the feedback from you if you can find this sort of pattern you know, in your kids, of course. And from here we will move to hyperexcitability and hypersynchrony, and a little bit about epilepsy. Now we have the proof that at least one of our mice had an epileptic seizure, and I will show you that. And finally, I will show you how the focal disruption of PCDH19 can actually lead about to a propagation of ectopic activity. So this focus behaves in a strange way, but this strange activity actually can propagate away in the brain following a normal pattern of connectivity. And I think that this might actually be important for the disease, especially in the light of some of the data that Paul has already showed you today. Finally, I will end with what are we planning to do next. We want to be totally, let's say, open to what we are doing in the lab, hoping that, you know, that might help starting collaborations and so on, which I think it's something we really do to the community of the patients, if nothing else. So, how the model has been designed? The model has been designed by taking one of the exons of the gene, exon 3, has been surrounded by two specific sequences. They are called LOXP sequences. They don't bring about any change at all in the final protein, so these mice are completely normal, but in presence of a special protein which is called recombinase, a protein that we do not express, that is not present in the mouse, is not present in our brain, is a bacterial protein. But if you add this protein, let's say with a virus, for example, the sequence, the genetic sequence that is in between the LOXP sites is cut out. So it's a way to actually do a sort of gene editing just by adding on top of the gene this particular thing. So in presence of this protein, recombinase, the cell that expresses recombinases loses exon 3. And exon 3 is still part of the is between the extracellular domain and the cytosolic tail. Most of the mutations actually that have been identified, this is an outdated diagram, but still shows clearly that most of the mutations are in the exon 1, but still there are mutations downstream of exon 3. What happens now when you actually kill exon 3? You can have two different outcomes. First of all, the production of a truncated protein, and second, but you could have the loss of the entire protein because there is, for example, a misframe construction of the RNA, which brings about to degradation of the RNA. It looks like, these are data from Silvia and Maria, these are real-time PCR data in cultured neurons, and it looks, there are probes designed against exon 1 and 2, probes against exon 4 and 5, and probes against exon 5 and 6. You can see that all of these real-time PCR demonstrate a drastic reduction in the amount of protein, and interestingly enough, exon 1 and 3 shows the maximum reduction. The reasons why there are differences is because when you do the various transfection, you may not have a 100% efficiency of transfection, quite obviously, but we think that we have a very strong reduction of the protein in this model. Unfortunately, as has been told by everybody, so I'm going to add my question, we have been totally unable to do after our in vivo experience, we've been totally unable to do immunohistochemistry against PCDH19, so we are not able to show the loss of the protein in our model. Our model is a bit weird, okay, but has some interesting advantages that may actually be quite important in the view of a novel notion we have about the disease. So the brain, as I told you, expresses normal PCDH19 except for the addition of these LOXP sequences. And the protein is going to be normal unless you adapt this external protein to the brain. And that is what we do. What we do is when the mouse is P0, so right after birth, we inject in one hemisphere a very tiny amount of a viral vector, an adeno-associated virus, that expresses recombinase, and expresses it also with a second gene, which becomes fluorescent only because of the action of recombinase. So basically, what we have, then we wait that the mouse is a young adult. We do experiment at 30 and 60 days after birth. And what we do, we take our mouse, we put under the 2-photon microscope, which is basically a very fancy, very expensive microscope that allows you to look at fluorescence in a living and behaving mouse. And we found out, we look if we can see fluorescence. If we can see fluorescence, that means that recombinase has entered in the brain and that acted on the genome. So basically, we have a very good representation of the fact that we actually have the loss of PCDH19, and we know exactly where the loss occurred because we can actually map the entire brain before the electrophysiology experiment. Then we go under in the electrophysiology setup, and we place 2 electrodes. One electrode is inside the fluorescent patch, so it is in the region that has lost PCDH19, and the other electrode is exactly in the opposite hemisphere in the same position. The reason why we have that will become apparent in the following. So basically, every brain has its own control, because one electrode records the electroencephalogram from the diseased area, and the other electrode records the electroencephalogram from a normal brain, which is not totally normal, as you will see, because there are connections between the 2 hemispheres, okay? And that will become important towards the end of the talk. Afterwards, we keep all the brain. We have now a very large collection of brains in our refrigerators, and we do immunohistochemistry. We tried in many ways to do the PCDH19 histochemistry without any success whatsoever. But we can actually, instead of looking at PCDH19, we can look for the recombinase protein, which is the protein that causes the recombination of the gene. So where we have CRE, where we have the signal from CRE, we can expect that PCDH19 has been lost in those neurons, but we don't have a direct proof of that. So these are slices of the brain, which we did the electrophysiology. This is the occipital cortex, so it's somewhere back here. We are in the visual cortex, and the reason of that because I know where the visual cortex is, so I've learned it at least after all these years, and also because you can use visual stimulation to actually provide a physiological input, which I hope will become important in the next year of experiment or so. So anyway, what you can see here, this is the cortex that received the injection. All the little dots are neurons expressing CRE recombinase. This is the opposite cortex. You see that there are no neurons. If we have a magnification of it, this is the primary visual cortex. The hippocampus is not infected, basically, but all the cortex seems to be infected. Only few neurons in CA1 are infected and so on. But the cortex, as you can see, has a nice, has received a nice dose of CRE recombinase. If you look at higher magnification, you see something interesting. So this is a transversal section of the cortex. These are superficial neurons, L1. This is L2, L3. Here is the beginning of L4. As we have seen before, most of the endogenous expression of PCDH19 is partially in layer 2-3 and in layer 5. And what you can see here is that most neurons, but not all neurons, in magenta we have the CRE recombinase. You can see that most neurons, but not all neurons, actually are expressing CRE recombinase. And here there is, the arrows point out to neurons that are completely devoid of CRE recombinase. And we have estimated that about 70, at the focus, in the central part, only about 70% of neurons, we have cross-stained that, of course, with neuronal markers. At the central part of the injection, we have about 70% of neurons that expresses CRE recombinase, therefore have lost PCDH19. But as we get closer to the periphery, actually the mosaic rate decreases. I'm mentioning this because, as we have heard, it is very important that we get a mosaic to reproduce the disease. So now, what about the physiology of this of these mice? What can we see when we do our recording? I mentioned to you the first thing I'm going to talk about is sleep. And I need to remind you some notions about sleep. Sleep disturbances are very common in most disease of the autistic spectrum. And I would say that most brain disease actually comes about with sleep disturbances. And I think this is quite important. But the other important fact is that sleep is not just something that makes our life just a little bit more bearable, but sleep, it is actually an essential mechanism for restoring the correct brain metabolism and for memory and learning. We would not have proper memory and learning if we don't have normal pattern sleep. Whoever has been sleep deprived, and I think most of us have been sleep deprived some part of our life, knows that the brain works in a different way in a state of brain deprivation. Sleep is actually very complex mechanism, and we know actually only little about how it works. We know that it's divided in phases, and one phase I'm going to focalize is called deep sleep or slow wave sleep. Slow wave sleep, I will show you lots of data about this, is a very important phase of human sleep, just like mouse sleep. Slow wave sleep, this is what slow wave sleep is. So in this experiment, we did a simultaneous recording with two different electrodes. This electrode is a local field potential electrode. So basically, this is very similar to what you do when you do an electroencephalogram, okay? So you record the activity of the population of neurons in the brain, not of a single neuron. But a single electrode, which is nearby the first one actually, is a patch lamp electrode. This is in vivo experiment. And this electrode has been targeted to a pyramidal neuron. So we can observe what a single neuron does during slow wave activity. Now, the reason why it's called slow wave activity is because when you look at the electroencephalogram, and that is true also for human, you see that the brain basically moves between two different states. It goes up and down, up and down, up and down. And these two states are characterized by the fact that, as you can see here, these two states actually modulate the excitability of the neurons, okay? And you can see that this single neuron actually can produce action potential, which has the vertical bar. So this is the neuron gets active and produces one bit of information. You can see that the action potential are not distributed uniformly in time, but they only occur when the trace is down. As I told you, physiologists really like to make life miserable to everybody else in the universe. So when the trace is down, the neuron produces action potential. So obviously, we call it upstate. Since it's down, we call it up. And when the trace is up and the neurons are inactive, obviously, we call it upstate. Okay, we call it downstate. So there is this reversal. The upstates occurs when the trace is down, and this is when neurons are active. And the downstates occurs when the trace is up and neurons are inactive. I'm really sorry for this. It's not my fault. It's not been this confusion has not been generated by me. But this is the terminology that is used. And you can actually tell that every single neuron has about 50% probability of producing between 0 and 2 spikes during every upstate. And we'll get back to that a little bit later on. So, another important fact is that slow wave activity, it is a way in which the brain synchronizes its own entirety. So this going up and down, it's something that occurs simultaneously in the entire brain. And this is very important from what happens before. So these are recordings from a normal brain. And this is one electrode on one hemisphere. This is another electrode in the other hemisphere. And I think you can actually notice that the up and down states are very nicely synchronized between the two hemispheres. This diagram down here, which we'll find it again, is called the spectrogram. It is a line with the traces up here. Time flows in the horizontal direction. In the vertical direction, we have frequencies. So that is a measurement of how rapidly this trace here is fluctuating up and down, okay? And the color is proportional to how much it is fluctuating every single frequency. And you can basically see that during the up states, so when the trace comes down, there is a very large increase in the amount of oscillations at high frequency. And this is what is represented by these flames here. And this is important when we'll analyze some data relative to hyperexcitability and epilepsy. Okay? So, why is that the brain is regulated between these two different states? This comes about, as we shall see, by the interaction between the two classes of neurons, which are the principal neurons and the interneurons. The principal neurons, as I said, they are responsible for everything we do, for the words I produce. They innervate our muscle, they produce all of our output, but their activity, and in green you have the principal neurons, is continuously controlled by a class of neurons that are called inhibitory interneurons, which get excited by principal neurons, okay? And when they get excited, they talk back to principal neurons, and these are the circle with inhibitory synapses, and the way they talk back, they actually try to turn down the activity of principal neurons. So it's like when we drive a car. I mean, our brain is a negative feedback system. When we drive a car and we turn around, we turn the wheel, but we continuously adjust the position of the wheel depending on, all the feedback, of the sensory feedback we have from the outside. So this is a continuous feedback mechanism. And the brain works in the same way. we have a continuous feedback between the principal neurons that only know excitement, okay, they're the Italian part of the brain, and inhibitory neurons that actually tends to turn down the excitement. Okay, let's not continue with the stupid, silly comparison, okay? So we have this continuous dialogue between these two systems. Understanding this dialogue is actually essential to understand many things, including sleep patterns and epilepsy. Why is that? Well, this is actually very recent data from a beautiful neuroscience paper, 2017, by the Giulio Tononi group. He's Italian, but has been in Wisconsin for most of his scientific career. And here you can actually see what happens during slow wave activity. Slow wave sleep is also called non-REM sleep because it's a phase that is different from REM sleep. So it's called non-REM sleep. Okay, fair enough. And here you can see the local field potential, which you can see it oscillates up and down, up and down. And here you can see that spikes, in this case, are concentrated, okay, are concentrated in down states. And these spikes are actually the activity of a specific, a very small class of interneurons, of inhibitory interneurons. They are the somatostatin interneurons, which actually are used to turn off the network during the down state. So there is a continuous dialogue between the two systems. Now, what happens in our model? So this is a recording from the dual, let's say, brain model in which we have the PCDH19 mutation side and the control side. So this is a recording from the control side, and this is the simultaneous recording from the side where has been injected Cre recombinase. You can see nice alternation between up states and down states. These are the up states, these are the down states. But you can also notice that the amplitude of the down states is a bit smaller in the hemisphere where we have lost the Cre recombinase. And we were very puzzled, but at the first experiment, we were very surprised by that. The amplitude of the up state was about 1/2. And we've been working on this sort of system for over 5 years at that time, and we never seen such a difference, but we said, well, this is n equal 1, so who knows, it may change. So we continue doing this experiment. Now we have done about 1 year of experiments on these models, and I'll skip this, and we actually found this difference in every single mouse that we have analyzed. So in this diagram, we have a measurement, it doesn't matter how it has been done really, a measurement of the amplitude of slow wave activity, okay, in 4 male mice, in 7 female mice, and these are the control. I will tell you what the control are. The males, of course, they've been injected with Cre recombinase, so the mosaicism is also present in these males because of what I showed you, that at most, we have about 70% of neurons that get infected by the viral vector that carries Cre recombinase, okay, at most. So also the males are mosaics, and we have never been able to produce with a viral vector a 100% mosaicism. Even if you get a 100% mosaicism, you would, at the border, you would actually lose lots of cells because the efficiency of transfection decreases. So as you can see, there is a massive loss of slow wave activity in the hemisphere, which has been injected with Cre recombinase. On the left, you have Cre recombinase. On the right, you have the control side. It must be said that the control side is actually a bit lower than the control, and we think that is because of the interference of the activity that comes from the injected hemisphere. Also, the duration of the upstates is actually lower in the injected hemisphere. That means that slow wave activity is actually decreased both in amplitude, in intensity, and in duration. What about the temporal evolution of slow wave activity? Well, there is a temporal evolution. So this is a normal mouse, and these are the up states and the down states. And what you can observe, this is data collected from 6 mice. As time passes by, this is about 40 minutes. of sleep, as you can tell, there is a slight decrement in the amplitude slow wave activity. And that reflects the fact that there is a decrement in sleep pressure. And that the same also happens in humans. In humans, at the beginning, you have very prolonged stages of slow wave activity. As time passes by, slow wave activity decrements and REM sleep actually becomes more relevant. What happens in the PCDH19 mouse after the injection? This is it. This is the data we saw before. You can see that there is actually a massive loss as time passes by. This is just 40 minutes. As time passes by, there is a massive loss of slow wave activity in the PCDH19 mouse. So what we would learn if we could transport this to humans is that they would have shorter episode of slow wave activity, and they would tend to be lost as the night passes about. You can also see that there is a disruption of slow wave activity also on the control side, possibly because of the interference caused by this hemisphere focal loss of PCDH19. There is more to that. And this is, I think, what it is a very interesting situation. This is a recording, and about 30% of our mice show this phenotype. This is a recording of the control hemisphere that still has low wave activity, as you can see, and this is the pre-injected hemisphere. So this is the PCDH19 disease brain. And as you can observe, the slow wave activity is completely lost in this side. You could think that this basically is an hemisphere in which PCDH19 either causes a waking up or the starting of a non-REM episode of sleep. From the electrophysiological point of view, if you just look at the brain signal, at the cortical signal, the two things are basically indistinguishable. Okay, so being wakeful or being in REM sleep would be more or less the same thing. But the other possibility is that the loss of slow wave activity, I told you, slow wave activity is made by the alternation of two states. Up states, lots of activity, down states, lots of inactivity, if you wish. So if we lose this alternation, it could be that we have complete loss of up states, so we are in a sort of perpetual down states, or we are in a continuous up state. So what's happening here? Is the brain getting calmer or it is getting more excited? Okay, and we can actually understand that. It's a little bit tricky, but the data is actually here. So what we have here, we take all the measurement we do, and we make a graph in which on the horizontal axis we have basically the amplitude of the oscillations we have. Okay, and this is the distribution of it. So basically, that is giving you an idea in time how the brain is oscillating and how much energy is putting in all the oscillation. This is what we get from this trace here, okay? So this distribution is very wide, and the reason is because we have low energy states, which are the down states, and which are on the left, okay? So the energy increases as we move to the right. So this part of this distribution is the low energy states, the down states. And on the right, we have the high energy states. So the question is, how is the PCDH19 mosaic side doing? Is it on the left, which would be low energy, so down states, a very inactive brain, or is on the right, which would indicate an awake brain? And this is how it is. You can see how it changes. It moves to the right, completely to the right. So basically, this is indicating that when we have the loss of slow wave activity, we are moving towards high activity. We are moving towards wakefulness. So recently, we were very happy to see this paper by Anne Poduri that was with us 2 years ago. She's not here, unfortunately, this year. And they actually did a questionnaire-based studies. So maybe some of you participated, as far as I know. About 50% of the patients actually reported disturbances of sleep. And as you can see, that they have to do with difficulty of going to sleep, frequent waking up, and so on. So why we think this is very important? It is very important because slow wave activity is fundamental, for example, for memory consolidation. If you lose selectively slow wave activity, you may even be sleeping, but if you selectively lose slow wave activity, you definitely are going to end up with a cognitive impairment. Just goes by that. Recently, with Renzo Guerini, who is a neurologist at the Meyer Hospital in Florence, we start evaluating EEGs that they had from patients that they kept during the night. In those studies, they were just looking at the epileptic phenotypes. All these patients didn't show any epilepsy during the night, but all these patients actually seems to be showing a very disrupted EEG, and with actually very small signs actually of slow wave activity. So we are just at the beginning of this study, but I think actually it would be very important to evaluate the sleep EEGs in patients. Do I have still some time? Yes. Okay, great. Now, I would like to talk to you a little bit about hyperexcitability, hypersynchrony, which is what we very often observe in these mice. So, as I mentioned before, in the mouse cortex, neurons show very little activity, very few action potential away from the upstates. And which means that the oscillation, the action potential, should actually be concentrated during upstate. But sometimes we observe something very interesting. So this is another recording from the control side is in blue, the PCDH19 knockout side is in red. You can see the slow wave activity has much smaller amplitude on the knockout side, but there is also something very interesting in this very recording that we are going to point out right here. So here we have isolated the high frequency oscillation of the electroencephalogram, which is something similar to what Paul has showed you before of his mice. And you can see that during this period in which we have, after this period, when we have normal slow wave activity, we have the starting of this inflection in the EEGs. And if you look more carefully to them, you can actually see that these are formed by very, very fast transition in the electrical potential. This is the indication of single unit activity. So these are single neurons that start firing. But as you can see, the firing should be, as I told you before, the firing should be concentrated exclusively during the upstates. But as you can see here, the firing proceeds independently if you are, this is an upstate, this is a downstate, if you are in an upstate or in a downstate. So this was one of the first clear indication that when the slow wave pattern breaks down, it goes into direction of higher activity, and in some cases, this is not epileptic seizure at all, but this is clearly a hyperexcitability phenotype. And actually, about 50% of our mice can show transient episodes only in the hemisphere that has been injected with crary combinase, can show transient episodes of bursting activity. This is another example. So this is the normal hemisphere in blue, but the crary injected hemisphere is here. And here you can see that there is an episode in red. Just look at the red trace. This continuous fluctuation are indication of this bursting activity. And another quite dramatic example is here. This is the control hemisphere in green. In blue, it is the hemisphere injected with crary combinase. And from this diagram here, this is the control side, this is the crest side. You can see this vertical bands is the up and down states. So it's a slow wave activity. So this hemisphere keeps sleeping normally, but the other hemisphere actually shows this ectopic activity. I want to end showing you this thing here. I unfortunately have to go out of. I decided to show you this after the conversation we had this morning. So this is in about 1 year of experiment. We actually found 3 mice dead in the cage, and they never die. Mice really never die. So we suspect that they may have had seizures. And during this experiment, something interesting happened. Didi, our postdoc, was a little bit, she was a bit tired, so it was tricky for her actually to grab the mouse. So the mouse was really stressed by, chasing it in the cage, and then eventually she did get the anesthetics, was a little bit less fast than it should have been. So the mouse was really very stressed. And as soon as the mouse received the anesthesia, this is what happened. Oh gosh. Okay. Why cannot I see it here? Yeah, looks like. Okay, let's see if we can play it here. Okay, that would be good enough for what we are. Okay, here it is. You can actually see the mouse starts seizing, and please notice that the seizure is strictly unilateral. You can see it is the left leg that is actually having this tonic-clonic activity, is the back leg, and the other side seemed to be absolutely fine. This mouse has been injected on the right, so this would be exactly a situation you would have with the seizure would be limited to just one hemisphere. And so I know this is just anecdotal evidence, but I want to point out something, that the clear seizures are actually very rare. They are very rare, also in patients. They might be very dramatic, very important, but it's not a phenotype, thanks God, that presented continuously. So it's not surprising that in mice we don't see it all the time. If we would see all the time, all the mice we have in the lab would be dead, basically, okay? So maybe in special condition we would see it. But the kind of activity we showed you before, as I told you, we can see it in 50%, at least in 50% of our mice during a 2-hour recording. And that activity would have lots of implication on cognitive processes, because neurons, when they are in this burst, they cannot process normally the word because they are doing something else. They are following this exogenous rhythm, which is caused by the activity. Okay, so I will go back to the presentation just to close it. Yeah, I have other data. I mean, I think this is actually kind of important, and I will only take 1 minute. So this is the activity during a burst. In green, we have the activity in the PCDH19 side, but in blue, we have the activity on the opposite hemisphere. And as you can see here, the normal hemisphere, it is actually following the epileptiform rhythm we have in the opposite hemisphere. Why I think this is important? I think this is important also because of some data that has been shown by Paul. The fact that now we are beginning to see regions in which we seem to see cortical dysplasia. So there is the potential that if we have focal epileptic form activity from this region, this is actually going to propagate in regions of the brain which are actually more normal. So this might actually have a clinical importance. Finally, I just want to close with this. What about why we need mosaicism? Well, there is one possible explanation. I mean, I have no proof of that, but keep in mind that all the phenotype we have seen requires to be explained that there is an alteration between the excitation and the inhibition, okay, side. Let's say that here we have 2 different neurons. I'm using the same color code that Paul used. So in green, we have normal neurons. In magenta, we have PCDH19 knockout neurons. And these are interneurons. Interneurons receive the input from the excitatory neurons, and then they speak to the postsynaptic pyramidal cell, to the postsynaptic excitatory neurons, decreasing their activity. Let's imagine that interneurons have a slight preference when they form synapses for neurons that expresses normal PCDH19 protein. If that happens, we would have this situation in which there is an imbalancing between the inhibitory output to normal neurons with respect to the PCDH19. If every pyramidal neurons And so we actually have two networks, and one of the two is less inhibited than the other one. But if all neurons do not express PCDH19, we would not have this competition for the two forms, and therefore at that point the output from inhibitory neurons will be balanced between the two different populations, because there are no two different populations. This is a very simplistic and stupid mechanism, but actually in this way we can actually make hypotheses. We can actually test on the models that Paul has, that we have. So in that sense, I think we may have something useful. And well, I think this is it. Okay, so I'm chasing for the last slide. That is the people in my lab, and thank you very much.
I actually have one quick question. So do you feel that the pattern of hyperexcitability that you see is different than other genetic mouse models of epilepsy? And the reason why I ask that is, could this be a clue to the clustering nature that we see in the human phenotype?
Well, I mean, I don't have experience with every single models, quite obviously. But for example, we have lots of experience with mouse model for Phelan-McDermid, and which is the knockout of the SHANK3 protein, which is again epilepsy and autistic spectra. In that model, we have seen no disruption at all of slow wave activity, but we do see hyperexcitability, which comes about, for example, after visual stimulation. And again, that can be explained by a disturbance of the recruitment of interneurons from the pyramidal neurons. I really think there are many different ways in which you can interfere with the excitation inhibition relationship, and each of this method of doing it causes actually a slightly different phenotype. But at the very end, that must be hit. I think actually this model has a very strong phenotype. I mean, we were amazed how strong it was. If you look, at the proper angle, if you look at the proper things, the effect on slow wave activity is actually very, very strong. And the hyperexcitability, you have to look for it. And yeah.
Thank you, Gian Michele. I think it was very stimulating talk. This is a fundamental question I have, because we have seen talks from Paul Thomas and Wei about developmental, really created problem. Here you're creating another model which is acute, and obviously you see a quite exaggerated phenotype, which without going into detail, I would expect from acute disruption. But I would like your comment, and that can be a long one, so you can keep it short, because that might be a discussion for lunchtime and over, because it's relevant for treatment, it's relevant to the time of intervention and how we understand. So how do you reconcile your acute ablation of PCDH19 function data with the developmental problems we see early? And where do you find a middle road where we should look probably next to really to better understand where to intervene and when?
I'm so happy and not very surprised you asked this question, actually. Everything you say is totally right. And the first thing is that I would love to work with your model, to the Australian model, in order to do any kind of physiology we can think about. This is my first, excited answer. The second thing is that in epileptic study, we are used to exaggerated model. Just think all the pharmacological models of epilepsy. I mean, thousands and thousands of papers, and sometimes you can find actually something interesting from these models. Now, I can turn it around a little bit, because these developmental effects are obviously extremely important. The clustering is extremely important, but at the very end, what is defining the computation of the network is the pattern of synaptic connections, which is going to depend, of course, on the original imprinting, which is given by developmental effects, but also what happens during experience during the period of synaptogenesis. And in these models, the disruption, we do it at P0, when in the mouse cortex, you are still forming lots of connections. So the superficial neurons, layer 2 and 3, only start differentiating 4 or 5 days actually before delivery, okay? So those neurons at P0, when we do the injection, neurons in superficial layers have been there for only 3-4 days, and they have to form the dendrites, they have to form the synapses. So we may not be that far away from a window, a time window where it's actually important. The problem is that we cannot basically, and I have to be honest, we cannot do anything better than this. We are trying with in utero electroporation to move a little bit far older time, because we can actually transfect the progenitors 4 or 5 days before delivery, so at the time in which the neuron actually would differentiate. But I agree with you, we should talk about that at lunch. And I don't...
Maybe one last question from Paul. We do have a 45-minute Q&A session at the end that we can also do, but...
I'll make it quick, and I'm sleep deprived as well, so hopefully I'll remember your answer, Gian. So with your system, all the cells that receive the Cre virus, do they all express the Cre protein? In other words, is it a ubiquitous promoter, constitutive? And further to that, could you tweak the system by using excitatory versus inhibitory specific promoters to get different results?
Maybe using in this, let's say, first year of experiment, Cre is under the CAG promoter. So every single neuron, but not astrocyte, actually is expressing it. So it's a neuronal promoter, okay? And actually it's not CAG, I think it's HSyn, because it's neuronal, okay? So it is strictly neuronal. So astrocyte express normal PCDH19, provided that they express it. I don't know, and I don't care at this stage. We are actually thinking of doing, of targeting specifically Cre to pyramidal cell, and we can do it, of course, and the HSyn promoter actually is very much pro-pyramidal neurons, but we could also send it to interneurons to see what happens. Now, all your data, and also the data have been shown by others, the localization suggest that maybe interneurons may not be expressing lots of it. But I think it should be done, as a matter of fact. I think it really should be done, and we are going to do it. We already have the viral vector to do that. So next time, if, you know, we will definitely have those data. Yeah.